Friends, today I am discussing with you an interesting topic that is very commonly we deal with or we come across, that is about the classification of nerve fibers. This becomes very relevant in connection with the fibers, the various fibers which are carried to the brain or in the peripheral nerves, and their the functional and anatomical importance. I have here in this picture the Erlanger and Gasser, the Nobel Prize winners in 1944, and their work to classify the nerve fibers. I will deal with this in a separate slide along with my discussion. Coming back, I'm just posing a simple question. What is a nerve or a nerve fiber? Just to recap or memorize you, the nerve fiber is a bundle of axons of different origin. These are of different origin, packed and projected to reach the destination site. Destination site may be the spinal cord, maybe the muscles, maybe the glands, or maybe to the viscera or the smooth muscles, or so on, which happens in case of a peripheral nervous system. In case of central nervous system, they are named as fascicles or bundles that form the white matter in the central nervous system. Just to give you an example, the medial lemniscus. Medial lemniscus is the bundle which is originating from the medulla reaching up to the thalamus. Then after crossing the posterior column fibers, they form the medial lemniscus. That means it is a ribbon-like bundle which is going or which is ascending medially to reach the thalamus. That is the medial lemniscus. Now, after knowing these nerve fibers, just to say, repeat again, it is a bundle of axons and which is packed and projected. Now, what are the various packaging layers of the axons? If you are looking at this picture, we have the various packaging layers of the axons. Here I have two nerves. I have two nerves here. One nerve is a myelinated nerve, another is an unmyelinated nerve. So now this myelinated nerve, this is the myelin sheath there, this is the myelin sheath, there is no myelin here. They are wrapped in this greenish layer of connective tissue, what is known as a endoneurium. This is a endoneurium that covers both the myelinated and non-myelinated axons. And within this layer, you can just see there, this yellow and the green, you have a space that is called a endoneurial space. This endoneurial space contains a fluid because this fluid is essential for the transfer of signals either here electrotonically or at the node of Ranvier. So this contains the endoneurial fluid that is similar to cerebrospinal fluid. So all these bundles of axons are they form fascicles. This is one fascicle here. They are grouped together. They form bundles or the fascicles. And these fascicles, you can just see that are covered by this pink layer. This is a perineurium. So now this covers all these bundles of axons or endoneurial things. That means that is the perineurium, and these are connective tissue layers and having a large amount of a glycocalyx in there, and this is perineurium. And you have the blood vessel here. You can just see these blood vessels. They are projecting through this thing. Now these fascicles are placed or grouped. So you can just see one, two, three, four, or five fascicles grouped together or ensheathed in the another layer of connective tissue, that is epineurium. This is epineurium, and this is the epineurial uh, sheath. This is the epineurial sheath. So now you have a space in the epineurial sheath. So that forms again another layer. So that means we have three layers: endoneurium covering the axons, perineurium covering the fascicles or bundles, and epineurium covering all the fascicles together to form the nerve. So that means a nerve fiber, they say for example, a sciatic nerve contains these type of bundles. Some are sensory, some are motor. So all these things are put together, originating from the different areas of the uh, leg or limb. Now moving on, because this understanding these layers become important in terms of the nerve injury. Whenever we want to classify the nerve injury, it becomes relevant and important. Now, how the nerve fibers are classified? They are classified according to the myelination. One, if they are, if there is a myelin sheath covered, this is a myelinated axons, and if it is not myelinated, it is unmyelinated. So that means by the myelination, presence or absence of myelination, they are called, also they are also called as a medullated or non-medullated uh, nerve fibers. Then according to the function here is sensory, motor, or mixed. Usually sensory, you suppose if supply to an organ, the input, the output, and the whatever the input output, put them together. So that means there are afferent axons and there are efferent nerve fibers, afferent nerve fibers and efferent nerve fibers. So the afferent is similar to sensory, the efferent is similar to motor. This is a mixed nerve. Then both of these myelinated fibers or unmyelinated fibers, or these function wise, they have a conduction velocity. They are either fast or slow conducting nerve fibers. Both of them myelinated. Say for example, if I am taking the myelinated nerve fibers, the greater the thickness of the nerve fibers, the faster they are conducting. Similarly, but their conduction velocities will be to the maximum, they may go up to 120 meters per second. That case of the myelinated axons, which has the largest diameter is around 20 microns or micrometers. Then unmyelinated axons, again, they are also depending upon the diameter. Here they are proportional to the square root of the diameter. So that is the conduction velocity. According to the conduction velocity, again, unmyelinated slow fibers, unmyelinated fast fibers, myelinated slow fibers, myelinated fast fibers, something like that. You can club these classification, or you can keep this classification independently. Now, the nerve fibers can also be classified according to the tissue or organ involved. Just I am naming them here, either somatic from the body, somatic nerve, the muscular nerve if it is supplying the muscle, vascular nerve if it is supplying the blood vessels, or if it is supplying the skin, cutaneous nerve, if it is originating from the visual, that is rods and cones, the visual or optic nerve, then auditory nerve from the hair cells of the cochlea, or vestibular nerve, that is either from the otolith organs or the semicircular canals, the olfactory nerve, gustatory nerve, autonomic nerves, that means those who are supplying the autonomic nervous system, the visceral nerve, say for example, they are supplying to the different viscera of the body, maybe thoracic, abdominal, or anything. A splanchnic nerve, it is the It is supplying the abdominal viscera, the spatial visceral afferents, especially those spatial visceral afferents are performing spatial functions like chemo functions or other specialized functions of the region. So baroafferents, the baroreceptor afferents, the renal nerve, cardiac nerve, so on, so forth. You can list them even more than what I have listed here. Now, they in somewhere in 1935 or 37 is this thing they have published. Maybe in 35, these are the two persons. They performed experiments on a frog sciatic nerve, and by recording the frog sciatic nerve action potential, they found there is some difference, or the waveforms of the compound action potential is different. And then they classified the nerve fibers according to the peaks of the compound action potential recorded from the proxiatic nerve. They won a Nobel Prize for the discoveries relating to the highly differentiated functions of a single nerve fiber. Okay, this is one of the part of the functions of the single nerve fiber, because they went on this subject. They received the Nobel Prize in 1944. Having understood the Erlanger Gasser work, and their what they did, they recorded the compound action potential. Just I am briefing you about how the compound action potential is recorded. The compound action potential is recorded by placing the electrodes on the surface of the nerve fiber. Suppose this is the nerve fiber on the surface of the nerve fiber, and when you place a recording electrode on the surface of the nerve fibers, you have all the time, excepting you just don't think about this, the red, the one in the red circle. This is outside is positive, inside is negative. When there is a positive and negative, there is no change, or there is no drift in the electrical activity, because they don't, these electrodes do not detect any drift in the activity. So now what happens if I stimulate here? Once I stimulate, a wave of depolarization is produced. This is the wave of depolarization. Now, what is this wave of depolarization? This wave of depolarization is the change in the polarity of the membrane. The outside bit, it becomes negative, and inside it becomes positive, and outside it is negative. So this is what is called a dipole. Dipole is a unit or a structure which separates the two dissimilar charges by a very small or infinitesimally small uh, distance. So this definitely uh, is the dipole here. Now what happens? The polarity changed. The entire, if you are looking at this, the wave of depolarization is moving. Now this wave of depolarization moves from this side to this side. Here is the stimulus, because this I have not shown. As this approaches the electrode here, the first part of the electrode, that is the recording electrode. So the active terminal of this recording electrode detects a potential change, because in reference to this, it is positive, so that there is a movement. However, whenever we are recording, we make it sure that it will make a upward deflection. Suppose if I replace this, the change the recording places. If I place this one here and this one here, then this would go just opposite. That's in the reverse direction. So to make ourselves comfortable, what we have done, or what is done is to make it upward deflection. So as this moves in, there is an upward deflection. You can see here. As this moves away, because you can just see this red circle along with this wave of depolarization, it is moving out. So now, as it moves away from the recording electrode, this is the active electrode. So now it completes one wave. So as it comes in between, almost it is reaching in between. So it becomes a flat curve, or it reaches the baseline level or isoelectric level. So now, as it approaches the other terminal, other electrode, because this is a active electrode here, then maybe positive. This is negative because the electrode should have a positive terminal and a negative terminal. As it goes to this thing, so the potential reverses and it makes a downward deflection. It's a downward deflection here. You can just see that. And as it moves away from the this second recording electrode, so it makes it a zero potential here. So this is the standard recording procedure for a compound potential. Now, in this recording compound potential, we have two waves. Say, for example, the one upward and one downward, a biphasic or a diphasic action potential it is called, and the amplitude of this action potential is in at the maximum in terms of millivolts, uh, maybe two millivolts or 3 millivolts or something like that. If you are looking at the amplitude of the real action potential, intracellular action potential, say for example, it will be the around 100 millivolts. So that means it is nearly 1/100th of the amplitude of the real action potential happening in the uh, nerve fiber. Suppose if I have introduced the intracellular electrode, then I could have got 100 millivolt deflection. That's the from the resting potential to the peak potential. Now, moving on, because the biphasic action potential sometimes they will because they will change the configuration of the things. So to avoid that, the crush is made at one end of the nerve. So this is the crushed nerve here. You can just see that how the monophasic extracellular potential or action potential is recorded. Again, this is the wave of depolarization. This box, as it moves to the A electrode. This is the active electrode. This is the active electrode, and this electrode is a ground electrode, or it is grounded to make it a zero electrode. So that is grounded in reference to the ground. This active electrode works. Now, as happens, it approaches to the A. The wave of depolarization approaches to the A. You get the potential here. You can just see that it's rising up. The number two phase, as it approaches, it reaches the peak. As it moves away from this electrode, it comes back, and as it moves here, it becomes zero and it becomes a flat curve. So that is what the basis for monophasic action potential. In the monophasic action potential, one end of the nerve is crushed, so that we will get the monophasic action potential. In case of a sciatic nerve stimulation, what happened? The other end, anyway, it is crushed or it is cut, so you will get this type of recordings. Now, so now what happens? So when you do that, you have this is a stimulating electrode. You have the recording electrode. So now, when you stimulate, so here I'm showing the strength relationship. That means it is a graded potential. At B, there is no response. At C, this second point, no response. Then at this point, when the stimulus strength is increased to a level little more, say for example, if I say that 1 unit, 2 unit, and 3 unit, when I have made 3 unit, it has increased a small potential. Okay, so now if I have made 4 units here, in this case the amplitude increased. And when I have increased to 5 units, you can just see that the signal stimulus artifact is increasing and the amplitude increased. So, and as I stimulate further, amplitude is increased and reached almost to the same height as this. Now, these amplitudes are the real amplitudes. These, in case of a compound action potential, these amplitudes represent the sum of the algebraic activity of all the nerve fibers, all the fiber activity present within this nerve. As I have mentioned, the nerve contains number of fibers, and it is the summated electrical activity of all that is recorded, and it is the recording of the wave of depolarization as it approaches to the recording electrode. electrode in reference to the active electrode. So this is what is happening. It's a graded response. Unlike the intracellular action potential, it is a all or none response. Here I am going to tell you, or I am trying to impress upon you that intracellular action potential is a all or none response. This extracellular action potential or a compound action potential is not all or none response. It's a graded response because at each stimulus, at increasing the stimulus, what happens? You have these wave of whatever the electronic potential strength increases, and it recruits a newer and more fibers. You can just see here, if it is producing a depolarization, suppose with the little thing, a small, the nearing areas are depolarized. Subsequently, more and more fibers are recruited, and you get the more activity. That is what about the graded response of the compound action potential. So now moving further, it is a just I am trying to give you the brief idea about the compound action potential. And the Erlanger and Gasser, they recorded the compound action potential, and what they found that when they stimulated the sciatic nerve, say for example, this is the sciatic nerve, they stimulated somewhere here. When they stimulated the sciatic nerve, they got the peaks like this, one peak, this is one peak here, then second peak here, and third peak here. So this is very clearly depicted here. So this is one peak, and this is second peak, and the third peak. And the Erlanger and Gasser, they named it as A, B, and C. Now in the A, so I don't know. They made it like that. And once after discovering A, B, and C, they found the sub peaks. The different other peaks are appearing, and these different peaks are appearing according to the conduction velocity of those nerve fibers. Say for example, this beta one is having a the slower muscle, slower than the alpha. So that means they appeared a little late, and the gamma the little later. So now I will come back with that little later. So now according to the conduction velocity, these peak appeared. So they nomenclatured as A alpha, A beta, A gamma. So now this is the A delta. The B fibers are only present in the preganglionic autonomic fibers. And in case of a somatic nerve like sciatic nerve, so you do not have these preganglionic autonomic nerve fibers. So now here you have A delta fibers, thinly myelinated fibers. So that corresponds to the B here. A alpha, beta, gamma, delta. B fibers are missing in the sciatic nerve or in the somatic nerves, as we see in the periphery. Or now, again, the large, slow conducting fibers, they appear late. They say, for example, this is stimulus artifact. They are appearing after 200 here in this diagram after 200 millisecond. So that means the latency is long. These are unmyelinated nerve fibers, and these are C fibers, A, B, C. Now, that is a little enlarged here. You can just see the amplitude of these potentials. It is in microvolts. That is, this is 1 millivolt, 2 millivolt, and 3 millivolt. 1000 microvolts is equal to 1 millivolt. So when it is magnified further, you can see the how these ABC peaks are shown. Okay. So now, after having been seen, now I want to mention here, say for example, if I place any, this is a stimulus is somewhere here, stimulus is delivered somewhere here, and if I place an electrode, I get only, because the all other peaks cannot be resolved, so it will be only as you see a single peak. Other peaks will not be there, they will be missing because they cannot be dispersed. Now, as I move the recording electrodes further in the nerve fiber, so the see, I am this is second place. In that second place, now the beta peak starts appearing. You can just see that beta peak coming up because its conduction velocity is now we can able to resolve that. Now, as I place the electrode in the third position here, if I I'm doing this thing, I'm able to see the another peak appearing, the A alpha, A beta, and A gamma. You just see that. And as I keep here the recording electrode in the last or almost last, I shall be able to resolve these one, two, three, and this is four. I'm just making A delta plus B. That means A delta or B, not plus in case of a sciatic nerve. And it is the C fiber. So that means you can just see that C fibers appears late, the B or A delta fibers in between, and alpha, beta, gamma come in sequence. So this is a some the classification of the nerve fiber according to the peaks. Okay. Now, having understood the classification, now let us see how that the various things, the fiber diameter and conduction velocities were calculated and classified. You look here, the type of fiber A, B, C. Now, A, B, and C. You can just see that. So A is divided into alpha, beta, gamma, and delta. And these alpha, beta, gamma, and delta have a diameter of about, the alpha fibers have a diameter of about 12 to 20 micron. I consider it as a 20 micron. And so then the next beta is 5 to 12. I make it half of that. So 20, 10, 20, 10. Then gamma, I make it, though range is 3 to 6, I make it 10 by half is 5. 20, 10, 5. Then I will come back here 2.5. So that is for easy remembrance, for our understanding. Suppose if I have a diameter, this is 20 diameter, that is the maximum there, that is alpha, beta is around 10 micrometers, 5, 2.5, and the C fibers between 1 and 2, or maybe I will just say 1 micron. So that is the fiber diameter of the things. Now let us see the conduction velocities. This is 70 to 120 meters per sec. Beta is 30 to 70 meters per sec. Then gamma is 15 to 30 meters per sec. Delta is 12 to 30 meters per sec. The B fibers, they are having a conduction velocity 3 to 15 meters per sec, and C fibers from the slowest 0.5 to 2 meters per second. So that is the C fiber, maybe dorsal root or sympathetic. Now, to simplify the conduction velocity, I will say here 100 meters if I take the alpha. Alpha I take 100 meters per sec. Beta I take 50, half. Then gamma I take half of 50, 25 meters per sec. Then delta I take half of 25, 12.5 meters per sec. Then that's about the delta or the beta fiber or B fibers. Then C fibers is about 1 meters per second. So for example, 100, 50, 25, 12.5, and 1 meters per second. That's how I give this thing. And here I have given the functions, these proprioception, somatic, motor, the muscle spindles or whatever. So these are alpha, the motor nerves supplying all the nerve extrafusal fibers. The touch pressure or A beta fibers. Then motor to the muscle spindles, the gamma motor neurons or the muscle spindles. These are the gamma motor neurons. A delta thinly myelinated fibers are they carry the pain, crude touch, thermal sensation. Those are carried by the anterolateral column, or they are called anterolateral column sensations. So then C fibers are pain, temperature, some mechanoreception, and other reflexes, the irritants. So these are dorsal root. The postganglionic sympathetic, postganglionic sympathetics, or the C fibers. This is about the Erlanger and Gasser classification. So, a group of physiologists who are working in the sensory physiology, they thought that whatever the Erlanger or Gasser has given, maybe they want to reform it So now they used the sensory nerve fiber classification, and they considered three aspects: one, fiber diameter, the conduction velocity, and the threshold. Now, according to that, they classified group 1, group 2, group 3, group 4 fibers. So now, when they wanted to mention about the threshold or the strength of stimulation, they used the suffix A or B. One A, one B. One A means their threshold is low. They are easily excitable. They require little strength. So one B is they require more strength. So that means a high threshold. So the low threshold, high threshold. Similarly, the group 2 fibers are also classified as A, group 2A, group 2B. So now this I have mentioned here, threshold is indicated by A or B suffixed to the groups. So that is the Lloyd and Hunter classification. And if we are looking at the Lloyd and Hunter classification and try to see group 1A fibers and group 1B fibers, they have almost a similar diameter. Only thing their thresholds, these are easily excitable, they require higher strength of stimulation. Group 1A fibers have a 12 to 20 fiber diameter. The 70 to 120, as I have already discussed, they are the muscle spindle or primary nerve endings. They are A alpha. Group 1B, they are coming from the Golgi tendon organs. They are also A alpha, but their threshold is high. Otherwise, the diameter is around 20 only. The conduction velocities are also similar to 100 meters per second. Then group 2 fibers, they range between 5 to 12 microns. I will just say half of this, that is 10 microns. 10 microns here, and the conduction velocity is 20 to 50. So that means I will take the 50 meters per second. They are for touch, kinesthesia, muscle spindle, especially the secondary nerve endings of the muscle spindle or group 2. And again, group 2A and B, they are coming there. These are secondary endings. So these are A beta fibers in terms of the Erlanger Gasser classification. So then group 3 fibers, the fiber diameter is half of this. That means 5 microns. The velocity is half of this, the 25 meters per second. These are gamma motor neurons, and 12.5 meters per second. These are A delta fibers. So already this is corresponding to gamma motor neurons, the pain, crude touch, pressure, temperature, A delta fibers. So this is about group 3 fibers. Then group 4 fibers are unmyelinated C fibers. So they carry pain, touch, pressure, temperature, etc. This is about the Lloyd and Hunter classification based upon the diameter, the conduction velocity, and the threshold. Having said that, the study was to see their relative susceptibility. Now, the first and foremost thing was when we apply local anesthetic on the nerve, what would be the susceptibility? Now, this is the degree or the gradation, the most susceptible and the least susceptible and intermediate. Local anesthetics block the C fibers. That means C fibers are more susceptible to local anesthetics. You can just imagine you have gone to a dental surgeon and wherein he has removed your teeth, but your touch sensation remained. Touch sensation remained, but the pain has gone. So C fibers carry the pain sensation. Local anesthetics abolish the pain sensation. Whereas the A fibers are least susceptible to this. So that means it remains. Suppose if there is a very heavy, very high dose of the local anesthetic, then that is a different matter altogether. But anyway, there is a numbness and there is some amount of sensation remains. The hypoxia, the B fibers, especially those fibers which are the thinly myelinated preganglionic fiber, autonomic fibers, they are these things. The pressure or the mechanical effect, the A fibers are more sensitive. So, for example, whenever you have slept with a odd posture, keeping your arm on the head, and then suddenly when you wake up in the morning and you see that you are not able to move your limb, so that is one of those these things wherein these particular pressure effect on the A type of fibers or the myelinated fibers is this is a sort of a neuropraxia or one sort of the first degree of the nerve injury. Anyway, when I cover the nerve injury, I will tell about that. But this pressure effect will be on the A fibers, and but the C fibers are least susceptible. You can just see that the hypoxia, pressure, and local anesthetic. It is BAC, intermediate ABB, and the least susceptible, the C fibers are least susceptible to hypoxia and the pressure, but the A fibers are least susceptible to the local anesthetics. Okay, then applied aspect I mentioned, local anesthetic abolishes the pain because they are sensitive to the local anesthetic. C fibers are sensitive to local anesthetic. Local pressure abolishes A fiber activity. An example I had given about Saturday night paralysis, wherein or when you have a pressure effect on the nerve that blocks that nerve conduction or nerve activity of the A fibers. Hypoxia influences the autonomic nerve activity. If you can just go back to the previous slide, then you will know it. Say for example, BAC. So that particular component. Now, so these are some of the books I had taken, and the it is the Candles Neuroscience, the guidance textbook of medical physiology, the international edition, Genong's review of this thing, the again the 210 edition, and I have taken it from these books. Some of them I have got it from the Nobel committees and a report from about the Erlanger Gasser uh, publications. Now, just I am trying to read about the assignments, and you can do it for yourself. To know what you have understood. Draw a label diagram of the compound action potential. That's one. Now mention the layers covering the axons. Classify the nerve fibers on electrophysiological basis as recorded by the compound action potential or like a Gasser classification. Classify the sensory nerve fibers. Now you try to look into the Lloyd and Hunter classification. Now, explain the following. Why touch is not lost after local anesthetic application on nerve fiber? You explain. Then, what is the basis of Saturday night paralysis? Look at that. Compound action potential exhibits graded response. Explain. I had given you the idea. B fibers are not seen in the peripheral nerve. Fill in the blanks. I would not give you a lead, and you try to do that. Group 1A, Group 1B have the same conduction velocity but differ in dash. Group 4 are dash fibers. B fibers are present in dash nerves. Endoneurium covers dash, epineurium covers dash, perineurium covers dash. Dipole is a structure that separates dash by infinitesimally small distance. Erlanger Gasser won Nobel Prize in Physiology for their work on just try to recapitulate. Okay. Uh, Thank you very much.